So, and then the intrinsic activity of the enzyme. So that could depend on the substrate effectors and phosphorylation state. So the catalyst itself can be changed, and a lot of the times it's changed by these effectors. These allosterically regulated enzymes have allosteric regulators. So for phosphofructokinase 1, do you know what one of those are? Allosterically regulated? I don't know if we covered it yet. But you know how we have 1,3-bis not 1,3-bis, not the 1,6-bis phosphofructose. You know, that's the product of PFK1. There's 2,6-bis phosphofructose is the allosteric regulator. And I think we talk about that in a little bit. Competing substrates, pH and ionic environment, and then the temperature, of course, will affect things too. So I think we covered all of that. And then rate of a reaction depends on concentration of substrates. And the rate is more sensitive to concentration at low concentrations, right? And that's because it becomes insensitive at high concentrations, just like I described for alcohol dehydrogenase. But at low concentrations, when you're like at the Vmax or below the Vmax, the Km rather, not the Vmax, half Vmax, which is the Km. If you're around the Km, any slight change will get a big boost or decrease in the rate, right? Does that make sense? And then I think we were talking about this too, right? Yeah. So this graph, like I just said, this shows you, this plots the metabolic concentration that you typically find in the cell, and then the Kms for those enzymes. And they're all fairly similar. So that means that most metabolites are in and around the concentration of the enzymes that's going to, you know, alter them. So the rates for those enzymes are going to vary greatly. They're not anywhere near the Vmax, half Vmax about. So phosphorylation of enzymes affects their activity also. I think we did this slide before too. Phosphorylation is catalyzed by protein kinases. Always remember protein kinases. Protein phosphatases reverse the action of protein kinase. They just hydrolyze that phosphate group off the protein. Typically proteins are phosphorylated on the hydroxyl of a serine, threonine, or tyrosine because they all have hydroxyl groups. And then there's regulatory proteins also that can respond to different substrates or phosphorylation to regulate those. And we talked about that with, what was it from chapter 14? Do you guys remember? There are 2 regulatory domains and then 2, you know, active domains of the protein. I think the regulatory domains come off, and then those active kinases can then diffuse and phosphorylate. What was that, protein kinase C or protein kinase A?
I was thinking about,
you know, I can show you that picture again in a minute. And then proteins have a finite lifespan, right? Proteins, we make proteins, they're not around in our body forever. There's a degradatory pathway to get rid of them because they're going to turn over. They'll be altered chemically. They'll be reactive with oxygen radicals that are products of metabolism. So they're not going to be around forever, and these are your average half-lives and days of different proteins. Liver's 0.9, kidneys 1.7, heart's 4.1, brain and muscle about 10.7 in days, half-life. So that means for every 10.7 days, that protein, half of it that was made on day 1 or day 0 is gone after 10.7 days. So, and then reactions far from equilibrium are common points of regulation. So this would be like hexokinase or phosphofructokinase 1, the heavily regulated sites in glycolysis. Whereas this would be the isomerase and so forth, where really the velocity is only going to be governed by the concentration of the products and substrates to drive them, you know, to run forward or reverse. And this is that table showing us those. And one of the big regulators is going to be ATP and AMP, right? Because basically the concentrations of ATP and AMP should be opposite of each other. When a cell has a high energetic state, there will be a high concentration of ATP with a low concentration of AMP. And that AMP is really sensitive to burning of ATP, whereas ADP kind of remains constant all the way around. So we'll see ATP, like we just said, it's a feedback inhibitor of phosphofructokinase 1. AMP is an activator, allosteric activator of phosphofructokinase 1. Or anything that's going to drive energy burning pathways, AMP will activate that. AMP will inhibit anything that stores energy. Does that make sense? So when you think about regulation of metabolism in the cell, think about the energetic state of the cell and what those, like AMP, high concentrations of AMP would indicate that the cell needs more energy, so you need to burn more fuel. So it's going to activate glycolysis or lipolysis, glycogenolysis, or you know what I'm saying? Whereas ATP will inhibit, if you have a high concentration of ATP, cells in fine energetic state, and it's time to just store molecules for energy rather than trying to make more and more ATP. Does that make sense?
The book references the epinephrine with the glucagon.
Yeah, so the epinephrine and glucagon are extracellular signals, and they're going to regulate phosphorylation states of different cells.
It says that, like, once it reaches down to the AMP, that's when you start the activity, and it sets up the ATP, then it activates and activates.
Yeah.
So is that referencing the concentration of the ATP?
So the epinephrine and glucagon are like, they're controllers of metabolism on like a body-wide.
Right.
Whereas like AMP concentrations are just locally in the cell.
Okay.
So, and there's different kinases that'll be activated by these or phosphatases. And at the same time, there's a kinase that's responsive to AMP concentrations, and that's this kinase here, right? AMP, what's it called exactly? AMP activated protein kinase, or AMPK. So high AMP and low ATP will activate AMP protein kinase, as will some hormones, right? They all will activate this AMP activated protein kinase. And an AMP activated protein kinase will decrease insulin secretion. Because insulin is the hormone in your body to tell you to store energy, get glucose out of the blood and just store it. So it activates pathways such as gluconeogenesis or not gluconeogenesis, but glycogenesis, or synthesis of glycogen, basically. So it'll inhibit fatty acid synthesis and cholesterol synthesis, because AMP activated kinase, that generally is activated with low energetic state, so you don't want to store energy again if you have a high concentration of AMP. It inhibits fatty acid synthesis and lipolysis in adipose tissue, and at fatty acid oxidation, it'll increase in glucose uptake and glycolysis in the heart and the brain. And then the food, it'll actually send an increase in food intake through the hypothalamus. So some enzymes in the pathway limit the flux of other metabolites more than others. So enzymes that are far from equilibrium are the ones that are regulated. So this repeating theme, if it's far from equilibrium, that's where you have the highly regulated steps. Not all regulated enzymes have the same effect on the entire pathway, and that goes into the hexokinase and phosphofructokinase 1. Phosphofructokinase 1 has heavily regulated, whereas hexokinase, are not totally, dedicated for glycolysis. That's just going to trap glucose into the cell, basically. Others regulate steady state concentrations of metabolites in response to changes in flux, and hexokinase, phosphofructokinase are appropriate targets for regulation in glycolytic flux. Increased hexokinase activity enables the activation of glucose, and increased phosphofructokinase 1 activity will enable the catabolism of the activated glucose. So you can activate glucose and you have this activated glucose, which is the product of hexokinase activity. It can be sent through glycolysis to burn for sugar, but that you also have to activate glucose in order to store it as glycogen. So this glucose coming from the hexokinase can be shuttled to glycogen metabolism versus glycolysis, depending on the state of the body and the extracellular signals that it's receiving. Whereas when you activate phosphofructokinase 1, that sugar is bound for glycolysis, right? So, and then hexokinase affects flux through glycolysis more than phosphofructokinase. And this is hexokinase 4, right? Did anybody know the difference between hexokinase 4 and hexokinase 1? Yeah, hexokinase is the same thing as glucokinase, right? Well, hexokinase 1 I think is found in all tissue. Hexokinase 4 is found mainly in the liver. And do you know anything about the Km between the two?
Are they opposites? Like one's really high?
Yeah, hexokinase 4 has a really high Km, or glucokinase has a really high Km, whereas hexokinase 1 has a really low Km. Does that make sense? So which one, so in the liver has the glucokinase. So it has a really high Km, so it's in the liver because that's where you store and basically distribute glucose to the rest of the body. You store glucose as glycogen there, so you're going to have a higher concentration of glucose there, and that's what it's used to working with there. So, or phosphohexosisomerase. So, and then the flow of glycogen synthase is controlled by glucose uptake and phosphorylation, right? So this is what I was going to say. Once you phosphorylate glucose with hexokinase, it can either be burned, right here, this can either be burned for fuel, or an isomerase can transform it to glucose 1-phosphate, and then it can be stored as glycogen. Once you go from glucose 1-phosphate, you have to uridanylate it, which activates it further for glycogen synthase to work. And we'll look at that mechanism here in a second. So the elasticity coefficient measures responsiveness of the substrate, and it's just where on the curve you're looking at. Like I said before, if you're below the Km, it'll be really responsive. The rates of change will be really responsive to the concentration of the substrate. Likewise, if you're at Vmax, enzymes are saturated, you have a very low elasticity coefficient. So glycolysis versus gluconeogenesis. If you guys remember, it's basically all of the enzymes are shared except for 3, right? For glycolysis. And those 3 are the heavily regulated steps that are far from equilibrium. All the other ones are pretty much near equilibrium, so it governs which direction they go is going to be the concentrations of the substrates and products. So which ones are regulated? Do you guys remember for glycolysis? Yep.
Phosphofructokinase 1 and hexokinase.
Yeah, so those are the 3 regulated steps. And in the reverse, pyruvate carboxylase, or PEB carboxylase replaces that pyruvate kinase, and those are transversely regulated. And then fructose 1,6-bisphosphatase, remember phosphatases remove phosphate groups. That's heavily regulated, opposite of phosphofructokinase 1. And then glucose 6-phosphatase is heavily regulated too, but that's only found in 2 organs in the body. And where are those? The liver, huh?
The kidneys.
And the kidneys, yeah. And that's the liver will respond to glucagon, perform glycogenolysis, and then and gluconeogenesis, and then make sugar to secrete it into the bloodstream. And that's how it does that. That last step, it takes glucose 6-phosphate and just removes that phosphate, and then spits it out of the cell.
So is the fructose 1,6-bisphosphatase committed to gluconeogenesis?
Is it committed to glucose? No, it's one of the regulated steps, and it's actually oppositely regulated from this enzyme. They're like kind of opposite. So there's 4 isozymes of hexokinase, and this is what we were talking about. Hexokinase 1 is expressed in all tissues to different levels, whereas hexokinase 4 is only expressed in the liver. It's also known as, what was it, glucokinase. Functions to clear blood glucose at higher glucose concentrations for storage of glycogen. It's not inhibited by glucose 6-phosphate, so it can function at higher glucose concentrations also, and has a higher Km. So isozymes, we all know what isozymes are, do we? Maybe not. This is your first biochemistry class. So they're basically different enzymes that catalyze the same reaction. And they can be different from a number of ways. They can be completely different genes, or a lot of these enzymes are different subunits that are just mixed and matched together. So creatinine kinase, there's different isozymes for that you can test, and you have different creatinine kinase in your heart than you do your normal muscle tissue and your brains. So kinetic properties of hexokinase 1 versus hexokinase 4, and if you notice, half Vmax here, hexokinase 1 is really low, so it has a really low Km, versus glucokinase has a really high Km. And it looks like almost hexokinase is, what kind of curve is that? It doesn't exactly follow Michaelis-Menten kinetics, right? So it's
almost half of that, half left.
not titration, well, yeah, kind of similar to a titration curve, but it's a sigmoidal shaped curve. So, like if you remember, there might be some cooperativity there. Like from the regulation of hemoglobin. So hexokinase 4 is regulated by sequestration and transcription. So it gets regulated. The regulatory protein will actually bind to it and reduce its activity, but it responds to glucose transport in the cell, will increase the hexokinase 4 to be produced and kicked out of the nucleus to phosphorylate glucose. Regulation of phosphofructokinase 1. So this is the entry step in the glycolysis. Fructose 6-phosphate and fructose 1,6-bisphosphate are the reactants and products, right? And it's the committed step to glycolysis. While ATP is a substrate, it's also a negative effector. So do not spin glucose in glycolysis if there's plenty of ATP. So ATP is basically like a feedback inhibitor, even though it's a substrate for this reaction. High concentrations of ATP will inhibit it. Look at this, citrate, high concentrations of citrate will inhibit it also. Why is that? Why would high concentrations of citrate indicate a high level of energy in the cell? Does anybody know what citrate does? Or where do we find citrate? And what energy producing pathway? Yeah, the citric acid cycle.
So it activates a lot of ATP production?
Yeah, exactly. So if you have a buildup of citrate concentration, your citric acid cycle or your TCA cycle is backed up, might be saturated, so you don't want to send more pyruvate into that citric acid cycle, so you want to stop glycolysis at that point. And here's your allosteric regulator. So fructose 2,6-bisphosphate activates PFK1, and then AMP and ADP will also activate it, right? And this is the effect of ATP on PFK1. So low ATP concentration, we get more of Michaelis-Menten with a lower Km value, whereas higher ATP concentration, it lowers this curve, increases the Km greatly, right? So you'll need more fructose 6-phosphate there to get to get the equal amount of activity than you would if it was in the low ATP state. So regulation of phosphofructokinase 1 and fructose 1,6-bisphosphatase. So these are the two enzymes that work opposite, right? And so you would think that they kind of, counteract each other, right? So AMP activates phosphofructokinase 1, and at the same time, AMP will inhibit fructose bisphosphatase 1. So you don't want to be making sugar and burning sugar at the same time, so they have to be oppositely regulated, right? So again, ATP inhibits phosphofructokinase 1, ADP activates, and AMP activates, and then citrate again inhibits phosphofructokinase 1. So fructose 2,6-bisphosphate, and this is our allosteric regulator of phosphofructokinase 1. So it's not a glycolytic intermediate, it's only a regulator. It's produced specifically to regulate glycolysis and gluconeogenesis. So it's not an intermediate, it's not like feedback inhibition. This is like it's actually made in response to cyclic AMP, but it's almost like cyclic AMP in that it's a secondary messenger that regulates the activity of both phosphofructokinase 1 and it inhibits fructose 1,6-bisphosphatase. So high concentrations of fructose 2,6-bisphosphate inhibit fructose 1,6-bisphosphatase and activates phosphofructokinase 1. So it's like word salad, this part of the class, especially here coming up. So glycolysis and gluconeogenesis differentially regulated by fructose, and these are just the graphs. So with fructose 2,6-bisphosphate, we make this curve like this, where we increase or decrease the Km basically of PFK1, right? And the opposite is true here. So with fructose 2,6-bisphosphate here, fructose 1,6-bisphosphatase activity has a much higher Km. Does that make sense? So it's oppositely regulated. And how do we produce fructose 2,6-bisphosphate? We produce it, the enzyme takes fructose 6-phosphate, phosphofructokinase 2 produces fructose 2,6-bisphosphate, and then fructose 2,6-bisphosphatase 2 is the enzyme that converts fructose 2,6-bisphosphate back to fructose 6-phosphate. So if you notice, PFK1 and fructose 1,6-bisphosphatase kind of take fructose 6-phosphate and make fructose 1,6-bisphosphate. They kind of are oppositely regulated here. And as it turns out, phosphofructokinase 2 and fructose bisphosphatase activity are they're opposite functions contained on one bifunctional enzyme that is regulated by phosphorylation. So in this figure, we have phosphofructokinase 2 here, when it's not phosphorylated, is in the active state, whereas fructose bisphosphatase is inactive. Glucagon will stimulate the production of cyclic AMP, which will activate cyclic AMP-dependent protein kinase and phosphorylate this bifunctional enzyme. When the bifunctional enzyme is phosphorylated, the phosphofructokinase 2 activity is inhibited or inactive, and the fructose bisphosphatase 2 activity is active, and you get production of, no, not production, so you reduce the concentration of fructose 2,6-bisphosphate, which inhibits glycolysis, stimulates gluconeogenesis. In response to insulin, phosphoprotein phosphatase, again, phosphatase removing phosphate groups, removes the phosphate group from PFK2 phospho This word salad gets crazy. Removes it from this bifunctional enzyme and activates the phosphofructokinase 2 activity, which will increase the concentration of fructose 2,6-bisphosphate, which is then the allosteric regulator that does what?
Stimulates glycolysis.
So yeah, increased is going to increase glycolysis, right? Or glycolysis. So, and then similarly, the pyruvate kinase is the last step in glycolysis. It's regulated also, and a similar type of theme here. High energetic state, you're going to have a lot of ATP, a lot of acetyl-CoAs, because acetyl-CoAs, you take pyruvate. Before you enter the TCA cycle, you break that pyruvate down into acetyl-CoA and carbon dioxide, right? And so high acetyl-CoA concentrations may mean there's the backup into the TCA cycle. And alanine, there's enough amino acids. So, and then inactivated by phosphorylation in response to signs of glucose depletion and glucagon in the liver. So glucose from the liver is exported to the brain and other vital organs. So similarly, so remember, things that feed into the glycolysis or pro-glycolysis, energy producing, are going to be inhibited by high energetic state of the cell, and then activated by a low energetic state of the cell. So, and this is the regulation of pyruvate kinase here. So we have phosphoenolpyruvate, that step, pyruvate kinase catalyzes the conversion of that to pyruvate. And then ATP, acetyl-CoA, long-chain fatty acids will all inactivate that pyruvate kinase. And then in the liver, glucagon will activate protein kinase A, which will phosphorylate pyruvate kinase and keep it in the inactive form. And that's because what is glucagon trying to do?
Insulin to the cells?
Doesn't
glucagon show insulin to the cells?
Not insulin. Glucagon kind of works the opposite. Yeah, glucose is insulin, right? So when you'll secrete, your pancreas will secrete glucagon in response to low blood sugar. So in the liver, when it gets that response from glucagon, it doesn't want to burn any more sugar. It wants to just make sugar. It stimulates gluconeogenesis in the liver and kicks sugar into the blood system to bring that blood sugar level back up to normal. So two alternative fates for pyruvate. Pyruvate can be a source of new glucose, so we can make new sugar out of pyruvate. So we can store it as glycogen, and it can also generate NADPH via the pentose phosphate pathway. So if you guys remember, NADPH is the supplier of reducing equivalents for any anabolic processes that your cells will do. So pyruvate can be a source of acetyl-CoA, of course, and that'll go directly into the citric acid cycle, or that acetyl-CoA can be stored as body fat, long-chain fatty acids. That acetyl-CoA, well, you make the long-chain fatty acids out of acetyl-CoA units, basically. And acetyl-CoA will stimulate glucose synthesis by activating pyruvate carboxylase. And these are the two alternative fates of pyruvate. So pyruvate carboxylase will create oxaloacetate and then go into gluconeogenesis. Pyruvate dehydrogenase decarboxylates pyruvate and makes acetyl-CoA, and then it'll go to the citric acid cycle. And then likewise, feedback inhibition of pyruvate dehydrogenase complex. If you have a lot of acetyl-CoA, you're going to send pyruvate the opposite direction via pyruvate carboxylase to make oxaloacetate, and then store that as sugar. If you have lots of acetyl-CoA around, you don't want to store it as fat or burn it as fat. TCA cycle might be backed up, so you're going to take that pyruvate and then store it as sugar to activate gluconeogenesis. So the amount of many metabolic enzymes controlled by transcription. So, and then a lot of these genes are regulated by extracellular signals, such as, so insulin regulates hexokinase 2 and 4 for glycolysis, phosphofructokinase 1. These are all regulated by insulin signal. So the CHREBP activates transcription in response to glucose. So GLUT2 receptor, or not receptor, transporter will transport glucose into the cell. Hexokinase 4 will phosphorylate it to glucose 6-phosphate, convert that through several steps to xylose 5-phosphate, which then activates protein phosphatase 2A, PP2A, which will dephosphorylate CHREBP. Did anybody know what that stands for?
I was wondering where the name come from.
So CHREBP is response element binding protein. So this is the response element binding protein. This is our carbohydrate response element, I believe. So CH means carbohydrate response element binding protein. Does that make sense? So they kind of make it simple, but then when they give it to you, they just give you these. So the response elements are the actual sequence of DNA, you know, that are regulated by these proteins. The binding proteins bind, they'll recognize that specific sequence. But in this case, the CHREBP, the binding protein, will not go into the nucleus if it's phosphorylated twice. So it requires protein phosphatase 2A to remove phosphate in the cytosol, and then it'll get transported to the nucleus, where PP2A again will remove another phosphate and allow CHREBP to bind to the response element on the DNA and recruit RNA polymerase, and then you get transcription at that gene to turn whatever is being made there. So the FOXO1 activates transcription in response to insulin, and this is just another example of a regulating, you know, DNA binding response binding protein. And again, so you have insulin binds the insulin receptor, which, do you guys remember, it's a tyrosine kinase type receptor, which will eventually downstream activate protein kinase B, which phosphorylates the FOXO1. Phosphoprotein phosphatase will remove that, and then the FOXO1 will be destroyed, and you know, after phosphorylation will be destroyed via ubiquitin proteasome process. Whereas if protein phosphatase removes this phosphate group, it'll go and transcribe its genes. So protein kinase B will actually take that FOXO1 out of the regulation, so you won't get transcription of the FOXO1. And this figure here just shows you all the different binding sites of different regulators. Here's the FOXO1. It binds to the DAF2, which is distal accessory factor 2 binding site sequence of DNA. So, and these all transcription regulated factors. So glycogen's energy source stored mainly in the liver and the muscle, right? And you guys remember the structure of glycogen? You have a long chain. These are alpha 1 to 4 glycosidic bonds. And then glycogen phosphorylase will take phosphate and cleave one of these, right? To release glucose 1 phosphate, and then you have a glycogen shortened by 1 residue. So glycogen phosphorylase adds a phosphate, or it's kind of the analogous, it's kind of analogous of hydrolase type enzymatic reactions. If you want to remove a phosphate, you just put water there instead. Water comes in and replaces, and that phosphate's a great leaving group. In this case, instead of water, phosphate comes in and removes the glucose from that chain, okay? So you add phosphate across that bond. And that's how glycogenolysis, that's the reaction glycogenolysis has. So glycogen phosphorylase works on the non-reducing ends until it reaches residue from an alpha 1 to 6 branch point. If you remember, glycogen has alpha 1 to 6 branch points about every, 8 residues or so. And then debranching enzyme will transfer a block of 3 residues to a non-reducing end of the chain, and then debranching enzyme will cleave the single remaining alpha 1 to 6 linked glucose, and they call that a limit dextrin. If you've heard of that, if you remember, dextrins are often have the main chain bonds alpha 1 to 6, whereas here they just call it a limit dextrin because it's an alpha 1 6, and then debranching enzyme cleaves that remaining out limit dextrin. So this is dealing with the branch points in glycogen. So glycogen phosphorylate will cleave each of the single ends back until you get about 3 away from the branch point. Transferase activity transfers these 3 to the non-reducing end, and then glycosidase activity will cleave off this one remaining sugar. And then you have another unbranched polymer that glycogen phosphorylase will continue to act on. So if you notice, from glycogen breakdown we get glucose 1-phosphate, but in order for glycolysis to occur, we have to convert that to glucose 6-phosphate. So there's it has to be isomerized. So phosphoglucomutase will isomerize it, and basically it works. The enzyme has been pre-phosphorylated on a serine residue. That then this alcohol will attack this phosphate group and get the phosphate off of, hydrolyze it basically off of it, and then donate the other phosphate group back to that serine residue. And then it's ready to catalyze another isomerization. So fairly simple. So glucose 6-phosphate is dephosphorylated in the liver to transport out of the liver. And this is, which one does this? Out of the liver. So glucose 6-phosphate gets secreted into the endoplasmic reticulum lumen, and then glucose 6-phosphatase then removes that. And this is only found in the liver and a little bit in the kidneys, but mostly you can pretty much, the liver is the only organ that does this. Not the only, but still, mostly. So then that glucose will be transported out of the cells to increase blood concentrations of glucose in response to glucagon. So glycogen is synthesized by glycogen synthase, and glucose is activated for this synthesis reaction by uridadylating it. And we'll talk about that. So we make UDP glucose is the substrate for glycogen synthase. So we have to convert, glucose 1-phosphate to UDP glucose, and we'll see how that works in a second. And then this glucose gets added to glycogen on the non-reducing end. So this is how the glucose gets activated. You have one sugar phosphate. It attacks that alpha phosphate on a nucleotide triphosphate, and then it releases pyrophosphate. So there's not a lot of energy that does this, but then pyrophosphate gets cleaved into the 2 phosphates by an inorganic phosphatase, and that's a really fast reaction. So it drives this reaction forward, and then you end up with the uridadylated sugar, or uridadylated UDP glucose. It's uridadylated. Now this sugar is then activated for glycogen synthesis. So glycogen branching enzyme will then, once you've added to the non-reducing end via the UDP glucose, glycogen of course has branching enzyme which will add, it'll create a 1,6 branch point and then add some of the bases that it cleaves off. If you notice, it cleaved this this portion off and then added it 1 to 6, made a new branch point, and then these are the original glycogen chain. So glycogen branching enzyme cleaves a long straight section of the glycogen and then adds it 4 residues down, makes it alpha 1 to 6 glycosidic bond. So glycogenin starts a new glycogen chain, and glycogenin is the core protein that we see at the center of glycogen. So, and it has the glycogen covalently bound to it. So here you have UDP glucose. So this is the activated glucose for glycogen synthesis. A tyrosine on glycogenin will attack that and reduce the UDP, which is uridine diphosphate. And then another UDP glucose can come in, and this is the reaction that adds to the non-reducing end. And you do that over and over and over again, and you get the beginnings of a glycogen chain. And then this is the general structure of a glycogen particle. You have glycogenin at the center, with glycogen coming out with then branch points and branch points and branch points. And why do we want so many branch points on glycogen?
It stores up more energy.
Yeah, so we can harvest energy faster, more energy faster, because these are cleaved by the non-reducing ends one at a time. And here we have, like, I'm not going to count it, but number of places to pull that single glucose off. Whereas if we just had one linear, it would just be one, right? So control of glycogen breakdown. So glucagon and epinephrine signaling pathway starts phosphorylation cascade via cyclic AMP. So it activates glycogen phosphorylase, and the glycogen phosphorylase cleaves glucose residues off of glycogen, generating glucose 1-phosphate. So, and the enzyme that does this is phosphorylase B, and it is less active in this state, whereas phosphorylase B kinase will phosphorylate it in response to glucagon in the liver, or epinephrine, calcium, or high AMP in the muscle. High AMP is a signal of what?
Energy response.
Low energetic state of the cell, right? So in the muscle, if you have a low energetic state, you remember you store glycogen in the muscle too. Phosphorylase B will be turned on, and then phosphorylate to convert it to phosphorylase A, which is phosphorylated, and then it's active, and it'll start taking that glycogen apart and releasing sugar into the muscle. Remember, the muscle can't release that sugar back into the bloodstream, but it'll release it into the cytosol for burning, converting it into more ATP because you have so much AMP. So, and then the opposite of that is phosphorylase A phosphatase, and that removes the phosphates from phosphorylase A, converting it back to the less active phosphorylase B. So epinephrine and glucagon both stimulate breakdown of glycogen, and glucagon stimulates the breakdown of glycogen in response to low blood sugar. So it's to keep the body's homeostasis, whereas epinephrine stimulates glycogen breakdown. Why?
For rapid energy?
Yeah, rapid energy. So epinephrine is your fight or flight hormone. So it'll stimulate glycogen breakdown because you need a lot of energy to, flee or fight, basically. So, and this is the basic signaling pathway. So glucagon, they both stimulate GS alpha, which will will activate adenylyl cyclase, creating a 20-fold increase in cyclic AMP, which will activate protein kinase A, which will activate phosphorylase B kinase, which will convert phosphorylase B to phosphorylase A, and then AMP will increase the activity of phosphorylase A, and then will take glycogen and produce glucose 1-phosphate. I think we saw this before in chapter, 13? Bioenergetics chapter? Yeah. So control of glycogen synthesis. Insulin is the main hormone that's energy storage, right? So that you got high blood glucose, it tells your cells to take that glucose out, and it tells your liver to produce glycogen. So insulin signaling pathway increase the glucose import into the muscle and stimulates the activity of muscle hexokinase. Activates glycogen synthase also and increases hexokinase activity, which enables activation of glucose. And the glycogen synthase makes glycogen for energy storage. So that's what the insulin does. It'll increase the glucose uptake from the blood, increase the hexokinase activity, which traps that glucose in there and activates it for glycogen synthesis. So glycogen synthase is also controlled by phosphorylation, and the enzyme that phosphorylates it is glycogen synthase kinase 3, GSK3. So insulin will block GSK3 activity, which then at the same time it activates protein phosphatase 1 activity, which will dephosphorylate any glycogen synthase and turn it to its active unphosphorylated state. So glucose 6-phosphate, and insulin all activate protein phosphatase 1, whereas glycogen synthase kinase 3 is inhibited by the action of insulin signaling. So control of carbohydrate metabolism in the liver. So high blood glucose, you'll have an increase in insulin, which will cause more glucose GLUT2 receptors to fuse with the membrane, increasing the concentration of the glucose inside, which will result in glycogen synthesis. Insulin will then cause synthesis of hexokinase 2, PFK1 pyruvate kinase, which will lead to increased glycolysis. Insulin also increases insulin-sensitive protein kinase, which activates protein phosphatase 1, which activates glycogen synthesis, decreases glycogen phosphorylase, which is, the opposite of glycogen synthesis, right? And it activates phosphorylase kinase, which decreases glycogen phosphorylase, and so forth. Whereas low blood glucose has the opposite effect, basically. So, and then control in the liver versus the muscle. So epinephrine in the muscle increases glycogenolysis and increases conversion to pyruvate. Epinephrine also increases glycolysis because the muscles are what you're going to be using for fighting and flighting, so you need that boost of ATP there. So you need to burn sugar in the muscles. Oppositely, epinephrine in the liver increases glycogenolysis, the breakdown of glycogen, just like in the muscle, but it decreases glycolysis and increases gluconeogenesis. So in response to epinephrine in the liver, you're breaking down glycogen to spit out glucose into the blood, and at the same time, you're making sugar from pyruvate or amino acids that are glucogenic and spitting it out into the blood to deliver it to the muscle where you're going to be using that energy for fighting or flying. Whereas glucagon only is responsive in the liver, and the glucagon signal in the liver causes glycogen to get broken down and secreted in the blood, increase the amount of glucose 6-phosphate, which will then eventually be converted to glucose. And pyruvate will be converted to glucose also via gluconeogenesis. So that's the main difference between the liver and the muscle, is with epinephrine, we increase glycolysis in the muscle and decrease it in the liver. And in the liver, we increase gluconeogenesis with epinephrine or glucagon. And then this is a table, glycogen storage diseases, that I'm not going to go into.